I'm Ifor Williams from Emory University. I'm a faculty member in the Department of Pathology, and my research is in mucosal immunology. And I'm here today at the Story Booth to talk about what I think is probably the most important series of observations that my laboratory has contributed to the field, and that has to do with the role of a cytokine known as Rank-ligand driving the differentiation of M cells, which is a special type of phagocytic epithelial cell involved in antigen sampling in Peyer's patches and other inductive sites. And this story kind of evolved over a period of years and was the product of just good luck and Eureka moments. And my graduate students at Emory have played key roles in just doing wonderful technical studies and recognizing when they were onto something important. So there was a great amount of teamwork involved in taking the key steps to further this discovery.
I was working for several years looking at these structures in the intestine, and I was using some pretty nice microscopy systems to do that.
So the story kind of starts with work that I was doing with Rebekah Taylor, looking at various organized lymphoid tissues in the mouse intestine. And we were really more interested in cryptopatches and isolated lymphoid follicles, which are small structures that aren't as well known as Peyer's patches, and had only been really discovered and started to be described in the years leading up to when Rebekah was starting in my lab. And we wanted to use microscopy to better understand what the component cells were and what was driving their organization.
We had looked at the Rank-ligand deficient mice and noticed that they were not doing what they were supposed to do as far as their Peyer's patches and taking up fluorescent particles and things like that. And we were unable to locate any M cells in the epithelial layer covering those Peyer's patches.
We ended up studying this molecule Rank-ligand to look at the stromal component of these structures and see how that might be influenced, especially by the presence or absence of lymphotoxin. And Rebekah got the reagents and made this key initial observation that the pattern in which Rank-ligand was expressed within the Peyer's patches is where we first noticed this, was really quite remarkable and suggested a strong effect that might be conditioning the epithelium above the stromal cells expressing Rank-ligand.
I just had a very successful time looking at these structures, publishing, giving talks, and pretty much the last few weeks, the last few months of my time there in Ifor's lab was spent making this what turned out to be a great observation that carried on through many years.
That information got published in the Journal of Immunology in a paper, and Rebekah presented it at AAI meetings.
That was the beginning, and I was lucky enough to be able to pass the torch, so to speak, to the incoming graduate student. And the torch was very bright. I had a little hard time letting it go. It was a very exciting time. I could tell that the project was really going to bloom into something amazing.
The next student that came to my laboratory was Kathryn, and at that point we really thought it was worth looking at whether Rank-ligand might be absolutely necessary to make M cells. And the way to do that experiment was to get mice that didn't have Rank-ligand and see if that impacted their M cell formation.
I was very eager to learn some imaging techniques and get my hands on some microscopes. So after I spent some time imaging for the M cells in our special mice, looking for when they did develop or when they didn't develop.
It was not easy to get those mice. It was not easy to breed those mice to do the experiments, but we persisted. We found a way to do it. And lo and behold, when we did the first experiments, it was pretty apparent we were onto something. and that we were seeing a very strong effect like we thought might be occurring.
The big Eureka moment for me came after then I started treating some of our Rank-ligand deficient mice with recombinant Rank-ligand and returned the protein to the system.
We spent several years doing further control experiments and fleshing out the story completely.
I remember looking through the microscope and just seeing all of these M cells on all of the small intestinal villi, and it was clear that we could control cell development throughout the small intestine, and that Eureka moment really drove me to continue in science.
And I've gone on since their departure and graduation to involve other students in kind of further follow-up projects. But it's all part of a cohesive set of observations, I think, that has established definitively an important pathway as to how M cells are made and what their role is for mucosal immune responses.